Reproduction is a crucial moment in the life of each living organism. This process can be made thanks to the cell division, which happens in different ways in eukaryotic cell and prokaryotic cell. In fact, prokaryotic organisms reproduce themselves by binary fission, while eukaryotic organisms do that by mitosis and meiosis. In this presentation, we have decided to analyze sexual reproduction and genetic diseases, which are caused by error in the gene's transmission or by external factors.
Meiosis is the process with whom sexual reproduction occurs. It is composed of two phases, meiosis I and meiosis II. The process starts when the cell has completed the interphase and received the signal reproduction. The first phase of meiosis is meiosis I. During prophase I, DNA condenses to form chromosomes and the spindle starts to form. Nuclear envelope and nucleoli disappear, and as the chromosomes first become visible, they form a particular structure called tetrads. During metaphase I, tetrads line up at the cell equator and the spindle is completely formed. During metaphase I, the crossing over occurs. Crossing over is the reason of the exchange of genetic material.
During NFS1, tetrads pull apart and the chromosomes with two chromatids move toward the poles. During telophase 1, the chromosomes decondense and the nuclear envelope reforms. Meiosis II is similar to meiosis I. The only difference is that DNA duplication and crossing over do not occur. At the end of the process, which started with one diploid cell, the result are four haploid cells.
Genetic diseases are malfunctions that originate when errors during meiosis happen. There are three types of diseases: chromosomic, monofactorial, and multifactorial diseases. They can originate because of many factors, such as problems during DNA duplication or the crossing over, or because of external causes like radiation. Chromosomic diseases occur when there are more or less chromosomes than their normal number. One of the most common diseases of this type is Down syndrome, in which the problem is created by an extra chromosome 21. It means there are three instead of two copies of the chromosome 21. In 90% of the cases, the extra gene is contained in the egg cell, only 10% in spermatozoons. The trisomy 21 is called homogeneous when the third chromosome 21 is located in all the cells of the body, mosaic trisomy 21 when the extra gene is only in specific cells. People with Down syndrome are usually born by non-carriers. This means that it isn't always an hereditary disease.
Monofactorial diseases are caused by the mutation of a gene. The most common one is achondroplasia, better known as dwarfism. It is caused by the inhibition of the gene 4, which creates the protein fibroblast growth factor receptor. People affected by dwarfism have smaller lips because of the absence of cartilage. There are two types of dwarfism, harmonic and disharmonic, if the lips grow in proportion or if they don't.
These kind of diseases are caused by many factors, hereditary and environmental ones. There are many illnesses of this type, such as diabetes, asthma, osteoporosis, and hypertension. Diabetes, for example, is caused by the absence of insulin. This can happen because of two reasons. The first one is that insulin is not synthesized because the cells have difficulties to obtain the glucose to produce it. The second reason is that the protein channels are closed, and this is called insulin resistance.